to a membrane that contains proteins that have particular functions, in this case, transport functions. Okay? And we will also talk about an aspect that is related to a membrane potential, to a charge difference between the two sides of a membrane that is characteristic for all biological membranes. So yesterday, the vesicles that you have seen, they would be basically no charge between the two sides of the membrane. So it's still something very artificial. Nature would not use these type of membranes. Okay? So let's start now with just the permeability of a membrane. So if you take a lipid bilayer that doesn't contain any proteins, something that you have produced yesterday, then this membrane bilayer, it's not impermeable. It depends from the molecule. Some molecules can pass, like oxygen, carbon dioxide. They can just pass the membrane. They are a type of small hydrophilic. If you get basically molecules like water, they can still pass, but less good. So water can still pass. For many cells, the permeability of a lipid bilayer for water would be good enough. For some particular cells, like in the kidney, it would be too low, because the kidney need to produce a lot of urine, need to resorb a lot of water in the cells there. So you would need particular mechanisms to enhance the water permeability. But for most other cells, this is still fine. Or if you consider, for instance, like glycerol, it's a type of alcohol with 3 alcohol groups. It's a small molecule. It can still partially pass. Or if you think about ethanol, if you drink this, it would pass membrane. It would just go into the cell. Now, if you have molecules like glucose, sucrose, you see then the membrane is quite a barrier. It's still not completely impermeable, but quite good. Still, cells would basically use mechanisms as soon that, for instance, sugars come in to trap them, because due to the slight solubility, you might lose the sugar again, and that's something cells never would like to do, because once the sugar has been taken up, it should be used for energy production or for the basically formation of carbohydrates, right? But then, if you consider ions, the membrane is completely impermeable. So there is basically a barrier for sodium, potassium, that is so high in terms of energy that the membrane is impermeable. Okay? So you see, membranes are impermeable, but only for charged molecules, while membranes are permeable for uncharged. So if you look now for different criteria, you have basically 3 criteria that you can use to make a good guess whether a substance is permeable or not. You would need to consider size, polarity, and whether it's an ion or not. Small molecules are more permeable than large molecules. Polarity, a polar molecule would be less permeable than an unpolar. Ions would not pass at all. So if you have basically a large molecule with a high polarity that on top is charged, this would never pass. On the opposite, if you have an uncharged, unpolar, very small molecule, like oxygen, it can pass. Yeah? So whenever you want to ask yourself, can this substance pass or not, you just need to consider these 3 criteria and to get a feeling, if you compare it to another substance, which of those would be more permeable or high permeable. You can use these 3 criteria. Now, since membranes are impermeable for ions, cells can use this property, and they do so. Cells keep a different ion composition inside than outside. That's very important, okay? Which is something we actually hardly can mimic. Remember when we do reconstitution? The outside and inside so far was almost similar. So if you want to basically do it more physiologically, you would need to reconstitute in one buffer, then to move this liposome into a different buffer to come more closer to biological situations. It's often not necessary. That's the good news. Proteins, membrane proteins, and proteins in general are very robust. They can work even, under sodium chloride conditions. Also in reality, on one side they would see sodium chloride, and on the other side they would see potassium chloride. So inside of the cell, there's hardly any sodium, but there's a lot of potassium. And outside of the cell, you have a lot of sodium and very little potassium. Very drastic differences. And if you want to work basically with membranes under more physiological conditions, you need to take this into account. I typically say, if you want to remember all of this, I always say, consider where life actually started. Life started in the ocean. And if you taste the water of ocean, it's salty. And it's sodium chloride. So the life started in an environment with a lot of sodium, a little potassium, little magnesium, little calcium, and a lot of chloride. And since life started in this environment, it was continued. In our body, the water between our cells is the ocean water. So we carry the ocean with us. Wherever we go, we have ocean water between our cells. Sodium chloride. But inside of the cells, you have potassium chloride. So cells need always to keep this difference. And we will talk about how they do so, but that's very important. The salt composition inside of a cell is different from the outside. Okay. So now, sometimes we need to transport now substance, right? For cellular life, you would need to take up, for instance, material. So you need to transport things. You might also need to transport substances that are not permeable, right? That are hydrophilic or that are charged. And for that, membranes are equipped with transporters. And you can consider two types of, or let's say, transporters. Two types of transporters. It's maybe a type of channel where the substance can just pass through, or a type of, we call it, in this case, transporter. It binds and releases. So it would basically, on one side, bind and take it to the other side and give it free. Two different principles, channels and transporters. Let's have basically a look on these transporters, on these carriers. So they have basically a conformation of three different conformations. One is open to one side, then they can get into a so-called included phase, and they can open to the other side. So in reality, you would basically have a transporter that can bind the molecule, can swallow it, and can release it. That would be basically a type of mechanism. And these transporters work in both directions. And it only depends from the concentration in which direction. If you have a lot of the substrate on that side, the transporter will move in this ward. Sometimes it will also go there, but since there's this gradient, the transporter would always, in the beginning, move molecules inside until you have an equilibrium, where the transporter would either move a molecule from the inside to the outside or from the outside in. So we call this passive. It will basically allow the transport, but not against a gradient, only with a gradient for this particular molecule. And this basically enhanced the permeability of the membrane enormously, which is shown here. So for instance here, glucose, for the glucose transporter. If you have a glucose transporter, glucose by itself would be very slowly go into the cell. There's a high impermeability for glucose. But if you have this protein here, this carrier, glucose can bind and be basically transported, and this speeds up the transport of the glucose enormously. And since the glucose needs to bind and to be released, it actually behaves like an enzyme. For those that are basically working, this is a so-called Michaelis-Menten kinetic-like curve, because there is a saturation at a certain moment where all transporters have bound the substrate, and then they cannot work faster. So in the beginning, it facilitates until all transporters are saturated with the substrate and are helping to move the substrate across the membrane. Okay, so that was here the example, for instance, glucose transporter in red blood cells. No? Red blood cells, I told you, they have lost all the organelles, no mitochondria. So the only thing what is left is they can metabolize glucose. They can take up glucose and use glucose to make ATP with a process we call glycolysis, right? So that's why in our blood it's very important always to have a stable glucose level. Right? And you know that when levels of glucose are not correct, that this is very dangerous. If you have too little glucose or too high glucose, you need to be typically treated. So the red blood cells swim in a glucose solution in the blood. And they need just to take it up. And when they use the glucose, then of course there's a gradient. There's a lot of glucose in the blood, but all the glucose that comes in is used to produce ATP. So the glucose transporter here, it's a passive transporter that just makes this membrane permeable to glucose, that all the glucose from the blood can enter into the red blood cell and can be used for cellular processes in the red blood cell. Okay? In some cases, we would need an accumulation. We would need to transport material against the gradient. So we need not a passive transporter, but an active transporter. So we need to turn a passive transporter into an active transporter. How can we do this? So what solutions did nature found? One way is basically you use a light, the energy of light, to drive changes in the protein that trigger an active transport. This is, for instance, in some bacteria used. There are proteins that, for instance, can use light to pump protons, bacterial rhodopsin. Or you can now link your passive transporter with a machinery that can use ATP. So you have an ATP-driven transporter. Or you use a transporter that actually not only transports your substrate of interest, but can co-transport another one and can use the gradient of this to co-transport. Yeah? For instance, typically here ions like protons or sodium can be used because I told you sodium is a lot of sodium outside. So it will be spontaneously derived into the cell when you have a transporter. And this transporter can then use basically this gradient to, for instance, in this case, move a substance out. We call this type of transporter then an antiporter, but you could also have a symporter that takes with the sodium another substance in. Okay? So that would be, now we have entered the world of the active transporters. We have turned a passive transporter into an active transporter that not only allows the passage, but can accumulate, can work against the gradient. Okay. Here's one example how complex the situation can be. What you see here, it's an intestinal cell. So these are the cells that are basically the border between our body and our gut. So now we had some nice tea. I think there was sugar in. So I have filled my intestine now with nice sugar. So all the sugar would be here. And we want to take it up. And we don't want to waste any sugar molecule. Any sugar molecule that I have now, taken up should be actually entering my body. So you need an active transporter. So what you see is basically an active transporter that moves the sugar in under the use of an ion gradient, in this case in our gut the sodium gradient. So that would be a symporter, an active transporter. We call it also a secondary active transporter because it is based on a gradient, on sodium gradient, to drive the sugar molecule in, right? Now the cells basically will accumulate all the sugar, and any sugar that's here left will be still transported, so that now sugar is left here, but all the sugar is in the cell. But there, of course, it should not stay, because the sugar is also needed for other cells. I told you, the red blood cells also need the sugar. So we need now the sugar to distribute all over. So the sugar needs to leave again the cell. And there is basically here a passive transporter enough, because the cell has now high concentrations of sugar, and any basically a surrounding in the blood, for instance. As soon as the red blood cells use the glucose level falls, and glucose can leave these intestinal cells in the blood again to basically sustain the sugar levels in the body, right? But now we have created an additional problem, because with this Zim porter, there is something entering the cell, the sodium. And at a certain moment, there would be no sodium gradient anymore. So we need to get rid of the sodium. So you need another transporter. In this case, it's an ATP-driven transporter. We call it also a primary active pump because it uses directly ATP, or in bacteria directly light, to pump. So you need here a transporter which we call the sodium-potassium ATPase, a protein that will be later introduced to you. You see, so a passive transporter and two active transporters. And two different types of active transporters are needed just to take up nutrients like sugar, like amino acids. And you can imagine for each sugar, you need a sugar transporter. For each amino acid, you need an amino acid transporter. Yeah? So it's not that one amino acid transporter recognizes all 20 amino acids. So you need so it's a huge complexity already in the level of our intestine, just to get nutrients in. Okay, so now let's have a look for these primary active pumps, these ATP-driven pumps. You can actually group them into 4 groups, okay? Just take it as an information. You don't, it's more that you get a feeling, because maybe later you want to study one of those proteins in your career more in detail. Then you have a good start because of this lecture, and then you can go more in detail. So we have basically here these 3 types, or 4 types, I would say. One can say 3 types. So you see already from the look, this looks different from that, and that looks different from that, but they look similar, just from the architecture. So we have 3 types of ATP-driven pumps, if you want to say. So it's 3 different motors. How you can compare this? You can compare this with an electro motor, a petrol motor, and a diesel motor. They are all motors, but they all work a little bit different just to make movement. And this is the same here. You have basically 3 different types of motors that use a different principle, but in the end to do transport. Okay? So let's maybe go to a little bit. If we start with the first type, they have such a strange name called P-type ATPases. And that has something to do with the way they are working. So what you see here is basically such a transporter. It's embedded here with one part in the membrane, and it has a large cytosolic part. Okay? And this part here, the cytosolic part, is important for the use of ATP, and the membrane part, it's important for the transport of the molecules. And you can see you can even subdivide them. Some work as sodium-potassium transporters, others work as calcium transporters, others work as proton transporters, others work as heavy metal transporters to remove heavy metals because they would be toxic, but we are eating them with our food. But there's a protection mechanism. You see, these pumps would pump out heavy metals to protect us. Or there's even lipids. So even to this family, they can handle small, very small molecules, but also very large molecules. Very exciting, because we are puzzled by the fact, how can a protein that is designed to move an ion change into a lipid transporter? Very strange. And we have not fully understood this, but we are getting there. Okay, so let's have a look. I talked about basically here this structure, membrane part, cytosolic part. And you see even the cytosolic part has different domains. And what this pump is doing is the following. It takes ATP and transfers the last phosphate, the gamma phosphate, onto the protein. In the next step, it removes this. Why would this be a motor, right? I mean, so phosphoric acid, do you remember the charge of phosphoric acid? When it would be linked, for instance, to a protein? -2. Okay, so this is Thomas, the protein, P-type ATPase, without a phosphate, without phosphoric acid. If I get phosphoric acid transferred, what will happen to me? Charged! And all the other amino acids around here that I'm built of, they are also, some of them are charged. So if you transfer this to me, whoa, I will change the conformation. And if I lose it, I will go back. If I get the charge, I will. And I mean, two negatively charged, can you imagine? So we have here Tripta, maybe negatively charged. Close, she's linked to an amino acid negatively charged, linked to a not negatively charged. And I would be linked neutral charged. Do we want to link? Yes, we link. I'm neutral charged. But now I get these negatively charged. And I feel already the negative. Oh, I will take you even. Sorry. But you see how she changed? She changed with the whole part change here starts to change. And now you remove the phosphate again, and we will be together again. So in biology, we call this phosphorylation and dephosphorylation. And the enzymes that are doing this is a kinase and a phosphatase. So this protein in the cytosolic part contains a kinase and a phosphatase. So it's a fusion, an evolution. Two enzymes came together. One phosphorylating, one dephosphorylating. And they even were linked to a membrane part. And when they make this dance of phosphorylation, dephosphorylation, when they do this dancing, part of them are linked to the membrane parts. And I will pull on the membrane domains. And that will basically change the membrane domains to either bind the ion or to release it, to open to one side or to open to the other side. Okay? So this is a motor that just plays with phosphorylation, dephosphorylation on the protein to do movement. Okay. And that's why the name P-type ATPase, because for phosphorylation, the P stands, this protein is phosphorylated. So that's one type of motor, okay? And I'm pretty sure that Tripta goes a bit more, some details also, what interesting things you can basically study on those transporters. So here's just shown, this is the domain, the phosphatase domain, the kinase domain, and you have a phosphorylation domain, the part that carries the phosphate and where it will be added on and removed. And that will trigger changes here in these arrangements of the membrane part to allow the binding and release of ions. Now let's come to the ADC transporters. You see they are a little bit different. They also build of domains, like one membrane domain, another membrane domain, two cytosolic parts. And those are basically able to bind also ATP and to hydrolyze. But in this case, this protein will be not phosphorylated. So this protein works a little bit different. How should we do this? You want to assist me again? I do it, you know, where I mean, on the atomic level, it's of course a bit more complex, but just to give you the principle, okay? So you need to imagine, here would be a cytosolic domain, and above would be the membrane, one membrane domain. I would be the other cytosolic domain, we would be actually identical. It doesn't need to be the case, we can also be, actually, since we are different, we call it heterodimer, two different subunits, and then we have basically here the membrane part, okay? So we can bind ATP. And when we bind ATP, we do it in the way that I bind a part and he binds a part, so that we get actually, do you want to give me? Like this. And we can bind another ATP. So we need to do this. Then we come together. We can hydrolyze. we can bind. We can hydrolyze the ADP, we can bind. You see what happens? So we move. And this movement will change our transmembrane domains again. It will open and close parts of them, right? So sometimes, thanks a lot, sometimes I do it like that, no? ATP binding, no? So this is basically a different type of motor, using ATP binding and hydrolysis to change conformations. Okay, now the last type. So this is just illustrated here. No? ATP binding brings these domains together. Hydrolysis takes them away, a little bit like the way we have practiced together. Thanks a lot for that. Okay, now the last type here. That looks very different, right? It has basically, again, here a cytosolic part, a membrane part. The cytosolic part looks like it sticks into the membrane. And that works more like from an airplane, a turbine. So actually, this part can bind ATP and hydrolyze ATP. And what will happen, it will basically turn this light orange part into rotation. And this, of course, looks not symmetric. So this sticks out is like that. And you can imagine when this here goes around, it will hit you. So it will basically hit the membrane parts. They also will change the conformation due to these hits. So it's like a turbine. It's a motor, but working with a different principle. Using ATP to make a rotation, leading to transport by changes in the membrane part. Do we have an example? Yeah, here we have an example. So we have basically here the membrane part. We have here the cytosolic part. You see that both parts are even composed not only of one proteins, but dozens of proteins. So we call this is a complex, the cytosolic complex. That's basically the membrane complex, the V0 and V1 complex that assemble together to this type of rotational one. No? And we call this V-type because it has been first found in the vacuole of cells. But it's not only in the vacuole, it's also, for instance, in butterflies, in the intestine found, in kidney we have it, in the plasma membrane. But originally it was first observed in the vacuole of cells. Okay. So now the F-type looks the same. It's like a turbine. The only difference is it works in the opposite way. So instead of basically using ATP, it makes ATP. So it actually uses a gradient of ions to produce ATP. So that's basically a pump that you find in bacteria and that you find in mitochondria. We will come to this. So it works to the same principle, but reverse. And that's also something in nature. Many processes can work reverse, and it depends, of course, from the chemical pathway, whether it would be only in one direction or would be in the other direction. So typically, even ATPases that use ATP could in principle work the other way around, but under natural conditions, simply the gradients that are existing are not high enough. So typically, the ATPases are creating gradients, and only the F-type is using a gradient to make ATP. Okay. And you would also find it here in the chloroplasts. So that's an example from the plants, because also plants need to make ATP. Okay. Good. So now we have basically talked about the world of transporters. We have talked about basically active transporters. We have talked about passive transporters. We have talked a little bit about simple diffusion. Remember the three criteria? Is it small, the size? No? Is it polar, non-polar, or is it charged? But we have not yet talked about these channels, right? How do channels actually work? No? And what can they do? So let's go maybe into the world of channels. So the first thing is, what do they do? So one important point for channels is that they are helping to generate a membrane potential. And that's in particular important for the mammalian cells, okay? Also their evolution has a little bit went different directions for fungi, for plant cells versus animal cells. So let's stay for a moment with animal cells. So let's take a membrane. You have here a membrane, remember? Inside of the cell there's a lot of potassium. And of course when you have a potassium you need a counter ion. Typically you would think about chloride, but it's not so much. It's more negatively charged actually proteins that work as ions intracellular. But you have here quite high concentrations of potassium and outside not. So I have overdone it, no potassium on that side, a lot of potassium on that side. So now we insert a channel. And this channel would be selective only for potassium. What would happen? Okay, now we have a pore. Of course, since there's a lot of potassium on that side, no potassium on that side, exactly. The channel would allow the potassium to pass. Wow. Now we move an ion out that is charged. That has a consequence, because when we move a charge, we will leave something behind, a negatively charged, which in principle would attract positive charges. Okay? So what happens is basically you move out a positive charge, so you get an excess of positive charge on that side, but at the same time, you build up this electrical gradient, which would work against further diffusion, because this positively charged ion would like to go back due to these electrostatic interactions on the inside. And you get an equilibrium. And in fact, already very little ions of potassium that are moved out would build up a strong electrical potential of 70 millivolt around. And that's the so-called transmembrane potential. So every living cell has a transmembrane potential. In animal cells, this transmembrane potential is built up on a, typically on a potassium gradient. In fungi and plants, it's a proton gradient. It's a different situation. Yeah? But just that you know, for the animal cells, the channels are here very important in terms of contributing to a membrane potential. Okay, so what can we do now with this membrane potential? So all of our cells have this. If you would measure, and we can do this, across the membrane potential, all cells would have this membrane potential. Because most of them, they rely on these potassium channels. Okay? These channels would only let potassium pass. If you now have another class of channels, for instance here, sodium channels, that are normally closed, but you would open them, what would happen? When you open them, sodium would go in. And that would basically collapse your membrane potential. It would depolarize. And this is, for some cells, possible. It's possible for nerve cells, because they contain these sodium channels. It's possible for muscle cells, because they contain this. And we call this basically an action potential. That comes from the medical world. Because as a physicist, you would wonder, no? As a physicist, why would you call this action potential? I mean, here is a potential, here is no potential. I would say it's a collapse of a potential. But the medical people say, since this is an unusual situation, normally you would always have a membrane potential, and in some cells it can shortly collapse. They call it action potential. So this is a situation which only very few cells can basically do, due to the presence of these regulated sodium channels that can open and close. Heart cells, skeletal cells, nerve cells, and can be used basically for electrical signal transmission. Okay? So you see that nature plays now with these different ion composition inside of the cell and outside of the cell. It is used to generate a membrane potential, and this membrane potential is used by appearance and disappearance as a signal. Okay, good. And of course, you can now consider basically different, these regulated, the sodium channels, no? As a type of regulated channels. They are not always open. They're always open and closed. They change between the states. And you can basically, obviously, control some channels, whether they are open or closed. That can be, for instance, based on an electrical membrane potential. No? They can be basically when the and you have the normal membrane potential, they are closed. If the membrane potential collapses or is inverted, they open. That would be voltage-sensitive channels. Or you can have a channel that opens and closes independence whether it binds a signal molecule. That is, for instance, between our nerve cells and muscle cells. Yeah, we come to this. Or you can have mechanically gated channels. Like when I talk to you in your ear, you have cells that basically in the membrane you have channels that are sensitive to the mechanical stress on the membrane that is transmitted by little cilia, which are in our ear cells. And whenever they start to vibrate, then these channels can open and close depending on the mechanical stress. Okay, so here's one basically example where you have this, neuron that actually is in contact to a muscle cell, and when the neuron basically depolarizes, it basically is due to the opening of sodium channels. So you get basically the opening of sodium channels, a collapse of the membrane potential, even overshoot, so you get an inverse membrane potential. Then these channels close, are inactivated, and basically the membrane recovers, and you get the membrane potential back, and then you can get with the next, basically again, the next reaction, the next impulse, and you have the communication either between nerve cells or nerve cells and muscle cells. Okay. Now, basically here, when on that type, we call it a synapse. It's a synapse between the nerve cells and the muscle cell. These muscle cells, they also contain an example of basically ion channel, which would be this type of ligand-gated ion channel. So the nerve cells, when they basically are activated, they release a neurotransmitter. And this neurotransmitter is called acetylcholine. And this neurotransmitter is actually binding to the muscle cell membrane on a receptor there. And this receptor is a channel. This channel is normally closed, but when basically acetylcholine, the neurotransmitter is binding, it opens and lets allow ions to pass, in particular sodium, so that the muscle cell membrane also depolarizes. And that leads actually to the opening of voltage-gated calcium channels. A lot of calcium goes into the muscle cells, and that triggers the interaction of muscle proteins, tropomyosin, mysin, actin, to do the contractions of the muscle. So you see that these channels are typically used to control signaling events. Okay, that's shown also here more in detail. Here's the nerve cell, where basically you get a depolarization. In this case, the depolarization opens a voltage-gated calcium channel. The calcium goes into the cytoplasm. That triggers the fusion of vesicles that are filled with neurotransmitter, acetylcholine, that is released. And the acetylcholine is then here basically binding to the acetylcholine receptor, a sodium channel that depolarizes this membrane, that opens voltage-gated sodium channels. These voltage-gated channels, sodium channels, even open calcium channels again. So you have a very complex interactions of different type of channels that allow me here to move or to think between the different communication between neurons, right? Okay, so maybe now the last part, let's have a look how such an ion channel works. So it looks always in the drawings as if a channel would be a pore. That is strange, right? Because if it is a pore, everything what is in the size of the pore or smaller should pass. Okay, let's have a look. If you take potassium and sodium, just as an atom, or as an ion, sodium is smaller than potassium. So a potassium channel, no, I told you, animal cells have these constant open potassium channels that make a membrane potential, that are contributing to the membrane potential. But why do they not let pass sodium if it would be a pore? In a pore, everything what is this size would pass. So something is wrong on our drawings. It cannot be that there's a pore like that. Huh? But then the next problem is, since there must be something selective, how can then the process be fast? I tell you a little story. I don't know, you are much more younger than me. But when I was in your age, I went to the discotheque. At that time there was discotheques, was very fancy. Typically, I don't know how you do it here, typically on Fridays when, all the school was over or the lectures were over, then on Friday or Saturday night, there's even a movie, Saturday Night Fever, maybe you have watched. So then we went to the discotheques. No? And do you know, in the discotheque, before you even enter, what is there? I can show you. There were basically two men standing. Can you stand up here? 2 men. Yeah, you want to 2 men. Typically there were 2 men standing. Yes. And do you know how these 2 men looked like? Do you know how these men would look like? Look like. And I came, and they were watching at me. It's okay. And they were watching at me. And they would say, no, Thomas, you cannot pass. So you are the ion which is not allowed to pass. And then, a beautiful lady, they would look, yeah, she's allowed to pass. She can just go in, right? But you know what happened then? There was a long queue of people, because it took them so much time. Yes, you, yeah, you know, yeah, you. So if you want to be selective, it takes time. Thanks a lot. So to be selective costs time. But these channels need to work very, very fast. And have to be selective. So there's a dilemma. How can you be selective and being fast? No one understood. Because if it's a pore, already the size, no? So, okay, if it's a pore, it would be fast. But in the same time, the pore needs to decide you can pass and you not. Even if you are smaller, you should not pass. And the secret was, if you want to say uncovered, then the first structures, the real structure of these channels became clear. Okay? So I draw it very primitive. So typically you have basically, these channels are composed of subunits, of identical subunits, that form a pore. Yes, there is a pore. Okay? But this pore is basically narrow and has basically on one part inside formed a type of, we call it selectivity filter. Okay? And now there's one important point what you need to maybe just remember yourself. If you put sodium chloride into water, what would happen? Ions. You have the sodium and you have the chloride. Is this all what would happen? So no, I would have a charge. Maybe I'm the sodium, you would be the chloride, negatively charged. What is with you? You have forgotten all the water. So thousands of molecules of water around. What happens with the water? Because the water was used to disrupt actually the interaction of the sodium and the chloride. So, and what happens to the water? I need again here your help. You see, there come 2 water molecules are there. 2 water molecules. If you are free water, what would you do? be free water, okay? And what else would you do as a water molecule eventually with each other?
We will interact with each other.
And what do we call this interaction? Hydrogen bonding sometimes, no? Yeah, okay. So, and now here I'm an ion. And what is so special about an ion in water? There would be a shell coming. So can you come around me a little bit? Yeah, you make a shell. And I feel happy here. I feel very happy. And there's a lot of more water. You are free water. But you are bound. Oh, bound. You know what it means? Come with me, no? Yeah, you are bound. So this is my hydration shell. That's my hydration shell. I don't want to lose. I'm very happy with those, no? And the chloride lady who looks for some other water molecules, she will be also very happy with the water. Okay, so it's not only my size, no? I have here water around. So the size is not different. So I don't want to lose this water. Unless, and now we come to this channel there. You see the channel? Okay. And you is almost like a massage, no? Like a massage. But now, this channel can mimic this water. So eventually, I feel very happy here because the side change of the amino acids or the backbone of the gives me basically the same interaction that I would have with the water molecules. You see basically there, but it only works when this interaction here is as good as with the water molecules. So if you have now a smaller ion like sodium, would have basically water shell, potassium would have a water shell. But since I need to lose my water shell and I need now to get a massage from the protein channel, I need to be basically exactly having the same interactions. If I'm too small and the channel is designed for potassium, I might interact, but only on that side. But that's not good enough because I also want to have my other side in the interaction that I normally had energetically here with my water molecules. Okay? So if this is the case, I will would be happy here. I'm also happy here. I'm happy here. If I'm a molecule that basically goes here in and can only partially interact, I'm not happy here, so I will leave. I will never enter the channel. Also the pore is basically, if you want to say large enough, but in this part to enter, I simply, don't have the same interactions with energetically as I had basically with the protein, as I had with the water molecules. So here I feel happy. Here I only feel happy if I'm really the ion that should be transported. Okay, but this will not give any transport yet, right? Because the only thing what I would do all the time would be happy here, happy there. There will be no, I will never go into the never, because I'm happy here, I'm happy here. Maybe I make it once. I mean, you see it's a long channel. Maybe I make it once to the side. But why would I energetically, energetically it's shorter here. Out, in. So there's another trick that is required. And the trick is that basically there's a second binding site in the channel, which is as good. So I feel here happy, I feel here happy. I also feel here happy. I feel happy here again, because it's the same environment as I would have with my water molecules. Okay? So that means in principle I still would be trapped, either here, or here, or here, or here. But now, of course, there's more potassium on that side. Do we have another potassium? You want to be another potassium molecule? Okay, just come down. So normally, of course, you imagine I'm surrounded by my water molecules. You see he's also surrounded by the water molecules. Right? So he feels happy also here. So if you enter now, you lose your water. You feel happy here. No? If you feel happy there also, right? But you can also come back, you feel also happy there. But now you feel happy there again. And now I come in and feel also happy here. What happens now? Why? Yes, you are right. You have to move. Why? Exactly, because we have the same charge. So we have two basically places. Once he has entered, and once he has entered this place, a new ion can come in and will push out. And now you go. Wow, on the other side. Now the next one, and I will be happy here, and the next one comes, I will be pushed out. And now you can imagine that it's selective, but still fast. So it's these two binding sites that provide the same environment as the ion would have with the water molecules, with the hydration shell, that makes these channels selective, but also fast. Okay, thanks so much. That is basically what I tried probably to explain here in these foils, but it's maybe easier just to do this dancing. Thanks a lot for your contribution here. Cool. Okay, so that's why we have basically here two places which mimic the environment as if I would have maybe my hydration shell. And that can only work because these interactions are designed by the space, by the interaction, by the carbonyl backbone groups, that basically this is specific for an ion of a particular size. Okay, and if you consider also, if you then have ions with higher charges, not only monovalent cations, but divalent cations, you need basically to provide the respective environment that these ions can lose the water. So this is basically drawn a little bit once more, just what we played, okay? We have here our channel, this selectivity filter with two places where the ion is happy. The ion will basically enter, one will lose the hydration shell, will be happy here, eventually moves to the other side because it's also happy there. But once you have two binding sites basically occupied, then you get this push out, and then you get this conductivity. And normally you have basically on one side high concentrations, example potassium, inside very high, but outside very low. So these channels work in this direction. If you would change the ion gradient in the other direction, the channel would work in the other direction, right? So the directionality is given by the gradient of the ion, not by the channel. The channel can work in both directions. Yeah, so then it's basically the last thing. Now we have talked about channels that can be regulated, that can be basically closed and opened. But there's one subgroup of channels that can also have a third state, an inactive state. And that's, for instance, with our sodium channels in the heart cells, in the brain cells. So they can switch between closed, open, inactive, closed, open, inactive. And that's important that basically you get these typically, you know, action potential curves that when you go to the doctor, you see also, or when you see movies from hospitals and so, where you see the electrical heartbeats recorded. And the curves look like that without going into detail due to the ability of these channels to have not only two states, but three states. Yeah, that would be the finishing for this lecture. So what I talked about you was that molecule transport across membranes can be passive or active. Active can be based on transporters. We differentiate between secondary transporters, primary active transporters. Secondary active transporters use the concentration gradient of one, basically, substance to move another substance to accumulate another. We have talked about active transporters, typically three different families. They have different working principles, but do the same job. They accumulate a substance against the gradient. And we have talked about channels and why channels can be selective and still fast. Yeah, thanks a lot for that.